Hello, thank you so much for joining this virtual event. I'm Erin Edwards. I'm the head of services at Visikol. And today I'm going to be talking to you about using 3D cell culture models and high content confocal imaging to evaluate efficacy and to expose toxic liabilities in the drug development pipeline. So just to give you a little bit of background about our company and what we do, just so that you have some context for where I'm coming from with a lot of what we'll be talking about today. Visikol is a contract research organization. So we are focused on taking different types of tissues, whether they're 2D or 3D cell culture models or whole tissues, and transforming those tissues, those 3D models, into some sort of actionable insight through the services that we offer. So these services can range from traditional confocal microscopy to more traditional pathology and slide scanning, all the way to more advanced 3D cell culture models, which is what we'll be talking about today. And the basic way that this pipeline works for us is that we develop these 3D cell culture models or acquire these whole tissues, take them through different sorts of processes, whether it's running an actual assay or just labeling these tissues, imaging them through some form of imaging, traditionally confocal, and then taking them through a series of image analysis algorithms in order to pull out these actionable insights. So the question becomes, when we're starting to think about things in 3D, exploring biology in 3D, why should we use in vitro tools in order to expose toxic liabilities or understand efficacy of a therapeutic? Well, in comparison to a lot of in vivo models, of course, these models are much less expensive and they can be higher throughput. They minimize some ethical concerns. But what's really interesting is that a lot of the times they can actually enable greater translational relevance. So if you think about some of the differences between species in terms of things like the types of transporters or drug receptors that different species will express, these in vitro models become really valuable in that we can produce sort of humanized in vitro models and really get the closest to how a drug would respond in a patient. And then the next question is, well, why 3D cell culture? If you're set on taking these in vitro approaches, why move to 3D cell culture as opposed to 2D? Traditional 2D cell culture approaches are very simplistic. They can be very high throughput, and they certainly have their application. But cells in the body don't really exist in a 2D environment. There's cell-cell interactions, cell ECM interactions, some transport properties that are really only able to be recapitulated when you culture cells in some sort of 3D environment. And just to highlight that idea a little bit more, we have some example data here from a breast cancer cell line that was established from a resected tumor. And upon the initial pathological evaluation of this resected tumor, it was actually found that this breast tumor was estrogen receptor positive. However, when this was taken to a cell line and really extrapolated, it was found that estrogen receptor expression was lost. So you can see here the pictures in the middle. You don't really get much labeling for estrogen receptor. But when we take these cells into 3D cell culture, we actually see that estrogen receptor expression is once again restored as it was in the native tissue, that initial tumor resection. And what's thought to be behind this is that cell-cell and cell ECM interaction that regulates expression of a number of proteins, in this case, estrogen receptor. But why would that be functionally important? Well, especially when you're screening drugs, perhaps those that are targeting the estrogen receptor, this can be really important because you want to make sure that your system recapitulates the in vivo case as much as possible. So we did a simple comparison of a number of anti-neoplastic agents here. And I'd like to draw your attention in particular to fulvestrant, which is boxed in a dotted black box here. What we saw is that the breast cancer cells, this wood cell line, was much more sensitive to treatment with fulvestrant when it was cultured in 3D versus cultured in 2D. And this is a bit counterintuitive because you would think if cells are cultured in 2D, they're much more exposed to a drug, so you'd think that they'd be more sensitive. But of course, if those cells are not expressing the target of interest, they're not going to respond in the way that we'd expect. So just sort of an interesting case here, underscoring the importance of using 3D cell culture models in a way that allows us to recapitulate the in vivo microenvironment and actually see the sorts of responses that you'd expect based on those differences that you can see between 2D and 3D environments. But it's important to note that 3D cell culture is not really a one-size-fits-all solution. So as I touched on, 2D cell culture models can still be very useful tools. They're much higher throughput, so they allow us to run many more assays than can be accomplished with 3D cell culture models. A lot of the time-consuming portions of running these 3D cell culture model-based assays comes from actually evaluating the entire tissue. So rather than taking one optical plane, if you're using imaging to analyze your tissues, instead you have to take a series, maybe 10, 15 Z steps in order to really fully characterize those tissues. So while you get much greater relevancy, you sort of, is at the cost of throughput. And of course, these models can be a bit more expensive, but if you compare them to things like animal models or even clinical testing, we're still moving towards that lower cost and higher throughput range. So they sort of sit somewhere between 2D cell culture models and those higher order in vivo models, and really everything has its application somewhere along the pipeline. So the idea here is not to completely replace animal models, but to sort of stream things down so that we can get there much more quickly and effectively. I also wanted to touch on a little bit of nomenclature here. So you'll hear folks use different terminology for 3D cell culture models. We sort of use this catch-all term, 3D cell culture models, to explain any sort of three-dimensional aggregate of cells. Spheroids in particularly are generally reserved for stem cell contexts. For example, there's that spheroid forming assay in evaluating cancer cell stemness. But a lot of the times you'll see this term spheroid used more broadly for any sort of sphere-shaped amalgamation of a couple of different cells, maybe one or multiple cell subtypes. Then you'll hear the term microtissue used. A lot of the time, this is sort of designed to mimic more so the functionality of in vivo tissues. So it can refer to either cell culture models, so like a spheroid, or sometimes even excised tissues. So some folks will take tumor biopsies and cut them up into much smaller pieces and use those as quote unquote microtissues in order to screen their therapeutics. And the last category that you'll hear used a lot is organoids. These are much more organized amalgamations of cells, and it's usually, this term is applying to cells that have been derived from some sort of stem lineage. So it's really a long-term guided differentiation process that gives you this larger structure. So you hear a lot about iPSC-derived neural organoids is a very, very common usage of that term. But overall, when you're considering working with 3D cell culture models from a perspective of building these 3D cell culture models, there's a couple considerations that are worth thinking about. So first of all is the source material. You can use cell lines, primary cells, or even stem cells, and each kind of has its pluses and minuses. So cell lines are very easy to work with. They're pretty inexpensive, reproducible. But of course, there are some changes that occur during the immortalization process that make some of their applications a little less well suited. So sometimes the preference is to use primary cells. They're much more challenging. There's a lot of variability. They're more expensive, and you can really only use them once. You can't continually propagate these guys in culture in order to run multiple experiments on the same lot. And then there's stem cells. These are sort of much more challenging and take much longer to develop just due to the oftentimes long differentiation protocols that are required. So this usually puts them in a more low throughput category. And then once you've chosen your source material and you want to generate the 3D cell culture models, there's a couple of other considerations. The means of generating 3D cell culture models have kind of evolved over the years. One of the earliest approaches was the hanging drop approach, wherein cells are seated in a drop that hangs from a plate insert. So sort of an inverted case. But there's a number of other approaches that have been improved upon, making the technology much more accessible. For example, this magnetic printing, wherein magnetic nanoshuttles are used to sort of encourage aggregation aggregation. You can use matrix-based approaches, or my personal favorite is the ultra-low attachment plates. So these are U-bottom plates that have a coating on them that prevent adhesion of the cells to the bottom of the plate. Thus, the cells are much more likely to aggregate. So they're very easy to use, which again lends to the sort of more high throughput nature of this approach. And there's always new plates and approaches to generating 3D cell culture models that are in development. So a lot of technology here, so a lot of different approaches, and sometimes one approach is more favorable than another for getting different cell subtypes to aggregate. So one of the biggest challenges, especially when you use primary cells, is actually getting cells to aggregate. Sometimes you get, in the picture on the far left here, just little micro aggregates of only a few cells. Sometimes they're very irregularly shaped, as you can see on the far right. But what we're typically aiming for here is a nice spheroidal aggregation, as you'll see in the center. Now, when you're looking to evaluate these 3D cell culture models, you run your assays, maybe you're screening compounds, whether it's for efficacy or toxicity. There's a couple of different ways that you can interrogate them, right? You can use some standard biochemical analyses, some different types of assays, pretty low cost most times, and the throughput can be high depending on the type of assay that you're looking to run. But a lot of the time, you're not getting any spatial or cellular information. Similarly, you can dissolve the spheroid and use immunolabeling based approaches or other dyes and use flow cytometry to evaluate your cells. So you get some cellular information here, but again, you're losing this spatial organization of that larger 3D structure. So the next step up in complexity, and thus a little bit more expensive as well, is to use wide field imaging. However, this will only allow you to really characterize the cells at the periphery of your 3D cell culture models, just because your optics are limited in terms of being able to actually get good Z resolution. You're allowing so much out of plane light in that you're not going to be allowed to actually see each individual region of your 3D cell culture model as you image through it. So in order to get a little bit more perspective on the different regions of 3D cell culture models, a lot of folks will use histopathology. So you can think of this from larger tissue scale, where you're sectioning larger tissues into a couple of micron slices. A similar approach can be taken using spheroids, but you can imagine if a spheroid is only 100, 200, 300 microns, this histopathology approach becomes very tedious and very low throughput. It's very difficult to get a spheroid evenly bisected using histopathology, and then to get repeat sections in order to get some 3D context there. So ultimately, one of the really nice approaches is to use confocal microscopy. You're able to image deep into the tissues and really only be visualizing a few cell layers at a time. So the cost does go up, the cost per well, in order to conduct this sort of analyses, just because of the time that's required, and thus the throughput is a bit lower, but you get a lot more information out of it here. One of the challenges, though, about using confocal microscopy is that you are sort of limited in depth. So you can see from the image on the right, the top two rows here, we have a spheroid, and if you're going from left to right and continuing on the bottom row here, you can see that as you image deeper and deeper into the spheroid, you sort of start to lose the cells at the center of the spheroid. And the reason for this is that all of the individual cells and their components are sort of acting like lenses and diffracting the light as it passes through each cell layer, such that when you get to the center of the spheroid, you're losing a lot of light, and thus you're not able to see those cells that are at the center. So we've developed a number of tissue clearing reagents. These are commercially available through Thermo Fisher under the name Cytovista. And you can see that if you now take that same spheroid and you've cleared it and you look at that Z stack going deeper and deeper in Z, now you can actually see all of the cells throughout the entire depth of the 3D cell culture model. And this, of course, allows us to see more total cells. So you can see here from this graph, as we're going through every Z slice, we're getting many more cells that are actually able to be imaged And again, the reason for this is the ability of the light to actually pass through the entire cell. So the clearing reagents are a refractive index matching solution such that that incident light no longer gets scattered. Instead, it's able to pass directly through these 3D cell culture models. So by allowing for that more complete characterization, you can think of things like, if there's some sort of heterogeneous distribution of how cells are responding to a therapeutic, now you're able to much more effectively characterize that rather than only characterizing the cells on the exterior surface. So I wanted to dive into an example here of an important application of 3D cell culture models. This is liver toxicity and disease. So we've developed at Visikol a number of 3D cell culture models in order to study both liver toxicity and disease. These range from either monocultures of just hepatocytes or HepaRG cells to more complex co-cultures with other supporting cell types. So in the model that I'll talk most about today, we've used HepaRG cells, which are an immortalized hepatocyte line. So it's a hepatocyte progenitor. And these have been co-cultured with a mix of non-parenchymal cells derived from the human liver. So those non-parenchymal cells sort of support the liver function in a number of ways. So I'll go through briefly here each of the cell types that's sort of featured in this model. Of course, we have the hepatocyte-like cells. These produce bile. They sort of function to metabolize a lot of drugs. They store glycogen and lipids, and they comprise about 70 to 80% of the liver. Liver sinusoidal endothelial cells here are labeled with CD31. They form this sort of a highly fenestrated sinusoid network that runs through the liver and really facilitates that blood filtration capacity of the liver. Then we have Kupffer cells. These are sort of macrophage-like cells. They patrol those liver sinusoids and they incite an inflammatory response when appropriate. There are stellate cells. When these guys become activated in a lot of disease contexts, they actually start producing a lot of extracellular matrix, which can lead to fibrosis, especially in the context of some diseases we'll talk about in a few minutes here. And then there's some other supporting cell subtypes. Here I'm highlighting cholangiocytes. These form sort of these bile duct structures that help transport bile throughout the liver and out of the liver. And there's a number of other cells that we haven't really talked about here that are derived from this non-parenchymal population. But altogether, these liver 3D cell culture models enable us to ask a number of different questions. So one of the first things that we've looked at using these models is drug-induced liver injury. So the ability to evaluate toxicity resulting from different types of therapeutics. So in this case, we dosed those HepaRG non-parenchymal co-culture models with increasing doses of acetaminophen in order to demonstrate this principle of using imaging to evaluate toxicity in these 3D cell culture models. So the 3D cell culture models have been labeled with both a viability indicator, here shown in green, and a mitochondrial health indicator. So the viability indicator works by exclusion. So a compromised membrane allows the viability indicator in, and then it gets fixed in place. So the more green you see, the more dead cells. And you can see that as we increase the dose of acetaminophen, we get more dead cells. But what's really nice about imaging is you can start to layer some of these different assays on top of one another to start to understand a mechanism behind toxicity or therapeutic efficacy. So in this case, we used a mitochondrial membrane potential indicator because it's supposed that if a drug is cholestatic in its mechanism of drug-induced liver injury, you actually get accumulation of bile acid that can cause damage to the mitochondria. So here we do see there is some of that effect here with the dosing with acetaminophen, such that we see a loss of mitochondrial membrane potential with increasing dose of acetaminophen. And to highlight another aspect of these 3D cell culture models, the inclusion of immune cell subtypes in not just liver 3D cell culture models, but a number of 3D cell culture models has become increasingly recognized as important to effectively screening different types of therapeutics for either toxicity or efficacy. So in this case, we dosed with trovafloxacin with and without stimulation with lipopolysaccharide. And we see that we get a pretty dramatic shift in the dose response curve when we either stimulate or don't stimulate, presumably due to the immune component that's present within these 3D cell culture models. In addition to these imaging-based approaches, we can also layer on top a number of other non-imaging-based approaches, a lot of the time using the same spheroid well. So you can multiplex 4 or 5 different colors within a single spheroid, but then you can also take cell culture supernatants and analyze those for things like bile acid secretion, LDH release. We can look at reactive metabolites, urea production, the list goes on. So it's a really nice way to evaluate a number of different endpoints, especially when you might have this increased relevancy afforded by 3D cell culture models. And the last thing I wanted to talk about with these 3D cell culture models, particularly in the liver context, is liver disease. So not just screening toxicity, but more complex processes that maybe can't quite be screened in 2D. So non-alcoholic fatty liver disease is growing in prevalence in the United States and across the world, really. And it sort of starts with this abnormal lipid accumulation. And as those lipid droplets continue to accumulate, you get increased inflammation. That inflammation eventually activates the stellate cells that we talked about a bit earlier and leads to deposition of a lot of extracellular matrix, and this is termed fibrosis. And this ultimately leads to sort of an advanced cell damage state and a lot of scarring that impairs liver functionality. So there's currently no approved therapeutics to address that end-stage fibrotic state. So a lot of folks are looking to evaluate these compounds, but the current 2D assays don't really do such a good job of evaluating compounds in this arena. So we use those 3D cell culture models in order to study a number of the processes along the continuum of this disease. So we started out with steatosis, that accumulation of lipid in the liver. Here we used a much more simplistic 3D cell culture model, and I just wanted to call attention to that. So we used HepG2 cells, which is an immortalized liver carcinoma cell line. And the reason being is they can be much sort of more high throughput, just because you can continue to propagate these cells in culture without the need to continually rethaw primary material and worry about the lot-to-lot variation there. So this sort of brings down the cost of this assay at the expense of the sort of later stage evaluation of more advanced stages of the disease, but it allows us to get a good approximation, at least if you're looking to do some side-by-side comparisons. So it's always good to think about what's the most important endpoint in this phase of the assay. And you can always continue to build on that and really optimize what you're looking for in terms of cost, throughput, and in vivo relevancy. So here we use these HepG2 spheroids. We dose them with increasing concentrations of oleic acid, and you see that the amount of lipid per cell does in fact exhibit this dose response that we'd expect to see. Again, we can multiplex this with some different endpoints, not just these imaging-based endpoints. We can look at things like TNF-alpha secretion. So you can start to get a sense of that next step, that inflammatory step. But ultimately, if you're looking to evaluate disease, in this case fibrosis, those stellate cells that I mentioned earlier are really the key to allowing the recapitulation of that disease state. They're the guys that are going to get activated and deposit ECM, so they really have to be a part of the model in order to recapitulate that disease most effectively. So here we took the same, that HEPA-RG non-parenchymal co-culture-based approach, treated with TGF-beta in order to stimulate fibrosis, and looked at a number of different imaging-based readouts. So we looked at activation of stellate cells, here shown in green, these 2 green markers, PDGFR-beta and alpha-SMA. And we see that we get a great increase in the amount of stellate cell activation when we stimulate with TGF-beta. Likewise, those stellate cells they start to deposit collagen, here labeled either collagen 1 or pan-collagen to label multiple collagen subtypes at once. And we see that we do get increased deposition of collagen as well. There have been a number of proposed approaches to ameliorate this fibrotic state, one of which is to treat with some sort of ALK5 inhibitor. So the sample therapeutic, if you will, that we've used here is an ALK5 inhibitor, and we see that it does ameliorate fibrosis to a pretty significant extent. Similarly, we can multiplex these with other non-imaging based assays to look at things like cytokine production, ALT and AST, commonly used metrics to evaluate liver functionality. And we, again, see that when you stimulate with TGF-beta, you get some sort of fold increase in these different cytokine readouts. But when you treat with an ALK5 inhibitor, we get that amelioration of the disease state. So that's just one example of using 3D cell culture assays in order to sort of study a disease more effectively. But there's a number of other examples out there for this 3D advantage. If you're looking at evaluating drug delivery approaches, how well a therapeutic can penetrate a solid tissue, of course you need some sort of solid tissue in order to evaluate. You can't just treat a monolayer of cells. So here I have a couple images on the top left of a spheroid model that we've used to evaluate drug delivery in a 3D cell culture model. Then on the right here, you see evaluating cell infiltration. So very similar to evaluating drug delivery, as folks move more and more towards these cellular-based approaches to treating disease or evaluating the immune component of different diseases, understanding cell infiltration, in particular the immune cell infiltration, becomes an important question. So by using 3D cell culture models, we're actually able to do that. Again, if you just have cells on a 2D monolayer, you can't really look at what they're infiltrating. And some of these studies can be conducted using excised tissues. It just becomes a lot more low throughput. So having this ability to recapitulate that state, whether it's a disease state or the treatment state, using a 3D cell culture model is a really powerful tool. And then on the bottom left here, I've given sort of a slightly different take on the 3D approach, evaluating drug permeability in a blood-brain barrier model. It's sort of a less 3D cell culture model, but you do still have some sort of 3D context of looking at the transport of some sort of material in a 3D context rather than, and in a cellular-based model rather than in a traditional approach. So there's many, many other applications of this advantage of 3D cell culture models and assays, and I hope you take the opportunity to explore them a bit. I'd be happy to take any questions, and I look forward to following up with each one of you on any questions that you may have.